So today we're going to talk about systems biology. There are many definitions of systems biology. Here's one from Wikipedia where it's described as a biology-based interdisciplinary study that focuses on complex interactions within systems using a more holistic rather than reductionist approach to biological and biomedical research. When I see that definition, I'm reminded of a quote from Supreme Court Justice Potter Stewart, who once said, "I know it when I see it." So here's an image meant to sort of define the difference between reductionist and holistic approaches. So here's an image from a field where a game is being played, and if you just look at that field here, look at this image, you can see it's a foot occupied on a field. But when you expand the image and you see that there's a dynamic game being played. This is a game of American football where my favorite team, the Pittsburgh Steelers, are playing the hapless Kansas City Chiefs, and I don't know for a fact that they won this game, but I would put my money on it. Anyway, the idea here is that by seeing a larger picture, we get an idea of what event is happening here rather than just studying a foot. Systems biology is not a new field. It dates back several decades. So here's maybe the first systems biologist, Conrad Howell Waddington, who described the epigenetic landscape. And it was this idea that you could have a cell and it could go down one developmental lineage, a second developmental lineage, or a third developmental lineage, and that these lineages were largely stationary, and that complexes of genes here, or depicted over here, kept these states discrete and allowed them to be maintained. These are so-called attractor states. There were others that also preceded Waddington, like Ludwig von Bertalanffy, who described general systems theory in physics and biology in 1950, where he defined a system as a group of interacting activities. And your goal was really to understand the results from parts and the whole. The interactions were, by definition, dynamic. And he proposed a shift to open systems, which is a system that continuously interacts with the environment. And he also cautioned against analogies that are useless in science and harmful in their practical consequences. Prior to 1980, scientists really focused on studying physiology and disease processes. And that included systems approaches and modeling approaches. For example, Hodgkin and Huxley constructed a mathematical model explaining the action potential propagation. Or Dennis Noble modeled the heart pacemaker in 1960. But after the advent of molecular biology in 1980, scientists began to pull more towards the study of genes in physiology and disease. These studies produced precise, sometimes binary results, things like determining the cDNA and the primary amino acid structure of a gene. That was a defined binary result. Looking at the expression pattern of a gene could tell you about what it might be doing where and when. And of course, using genetics to perturb gene function in an organism by knocking it out or overexpressing it. And all of this is really compelling science, and it continues to this day. Walk the hallways at Penn or in any other major research institution, and most of the posters will be pursuing this style of research. So our first question: systems biology differs from standard molecular biology by A, focusing less on star players and more on interactions, B, leveraging data-driven science and integrative approaches, C, incorporating quantitative methods, D, all of the above, or E, just A and B. And the answer is just A and B. As we've seen in many other lectures this semester, many of the standard molecular biology methods, like sequencing, incorporate quantitative methods. It's not necessarily the incorporating quantitative methods that makes molecular biology different from systems biology. It's really the general philosophy to study the interactions and really consider the whole rather than a few key players or parts in the approach. So here are the goals of systems biology. First is defining the parts list of a system. Second is understanding how those parts are wired together, how do the pieces fit and work together. And third is describing the system quantitatively, and that usually means mathematically. So here's a system that's very well studied, modeling the lac operon. And if you look in PubMed, there are literally dozens and dozens of papers that quantitatively model the lac operon, and it works quite well. And that's because it's an extremely well-studied system with enumerated parts. The models that have been developed explain the observed behavior, and in fact, they even predict biochemistry and metabolism. They predict things that haven't already been measured. So that's when you really understand a system, is when you can predict things about it that you didn't already know. So in general, quantitative models of cellular networks try to be comprehensive based on bottom-up approaches, where they'll take biochemical data from multiple tissues and model systems. Data is typically sourced from the primary literature, in other words, reading papers. Where variables are missing, they're often estimated at or arrived at via permutation. And here's an example from a friend and colleague, Danny Forger, which was an older model, a state-of-the-art model with 74 variables and 36 reactions, where here's an equation that describes protein levels, here's another equation that describes nuclear translocation, here's a dimerization process of several of the key components, and here's the location of the dimer in the nucleus. So all these have quantitative algorithmic descriptions that are modeled in software. And what you can say about these quantitative models of function is that there's many variables. So transcription, translation, splicing, mRNA and protein degradation, protein interactions, how they're modified, where they are in a cell, nuclear versus cytoplasmic versus membrane, and of course, when they are functional. And one of the issues with modeling is that we have incomplete knowledge of most, if not all, of these biochemical parameters. A second question is, do you need to know them all to be able to understand something about a system? Maybe yes, maybe no. Another sort of critique of the style of modeling is that it's not necessarily designed to predict what happens following a perturbation. So it doesn't always predict what happens after you alter a gene's function by dosage, for example. And there are many of these types of perturbations. You can think about drugs as a perturbing agent or perturbagen. RNAi reagents can be perturbagens. Mutations to a cDNA or a gene can be a permutation. There are many physiological perturbations, for example, exercise or hypoxia. And all of these things that are occurring, we want to understand how they work, because that's really how physiology and pathophysiology works. It's a perturbation to a system. Another sort of criticism of the sort of bottom-up modeling is much of the data that goes into these models is hodgepodge at best, or severely underpowered at worst. And that means that the experiments and data collected weren't necessarily done with modeling in mind. I'll just sort of give you an example. For a particular process, you might have biochemical data that where parameters have been measured in cell lines. For example, a COS7 cell line, which is a green monkey cell line. And you might mix that with other biochemical measures that may come from a rat model, maybe from the rat liver. So you're taking different models, one in vivo and one in vitro, in different species, and trying to combine biochemical parameters identified in completely different areas, and make something sensible out of them. So our second question, true or false? Truly understanding a system with a quantitative model allows you to predict its behavior. And I think that's true. And what the modelers have shown us with really well enumerated systems and increasingly complex and predictive models is that when you truly understand something, you can predict its behavior. I'll give you another analogy, maybe the kind that Ludwig cautioned us against earlier, but I'll do it anyway. And that is, when you design a system, for example, when you design a car, you understand all of the parts that go into the car. You've made many cars before, and without even turning the engine on, by knowing the weight of the car, the approximate amount of horsepower that powers the car, the type of aerodynamics and the types of wheels that you have on the car, you can very accurately predict, for example, its 0 to 60 acceleration time at sea level, simply by understanding how that car is put together, what the parts are, and how it's wired. So around 2000, systems biology was rejuvenated. So there were several landmark studies, an increasing number of genome sequences, including ours, Homo sapiens, the sort of widespread adoption of genome-wide experimental methodologies like RNA expression dynamics using arrays, now sequencing, genetic screening where we have increasing amounts of sequencing data, and proteomic approaches. And so sort of in recognition of that, here's a partial list of departments and centers that conduct systems biology research. And you can see they're all over. It's a rapidly growing discipline. So let's talk about the first goal of systems biology, which is defining the parts list. I would argue that much of what we've learned this semester is really the tools that are principally used to define the parts list. And that includes genome sequencing. What are the genes? What are the cDNAs? What are the chromatin modifications? Transcriptomics, when and where are they expressed? Proteomics, also when and where are the proteins expressed? And we're going to hear more about this in a few lectures from David Spiker. How are they modified? Who do they interact with? Which we'll talk about today. Metabolomics, which is what types of metabolites are produced and how do they function. And there's an increasing number of omic sciences, so many that I have several friends who collect lists of new and ever more creative omics words. So the second goal, which is going to be the majority of this lecture, is understanding how the parts are wired together. And that really boils down to interaction mapping. So there are two leading approaches to defining physical interactions. The first is the yeast-2 hybrid or 2 hybrid approach, and the second is IP mass spectrometry. And these methods have been applied at scale to understanding interaction networks in yeast, elegans, Drosophila, Homo sapiens, and of course many other species. Of course, there are also many other types of interactions, and again, we've discussed many of those types in previous lectures. So I don't want you to think about this as being completely restrictive, and many of the following concepts will apply to those areas as well. So here's a version of the classical yeast-2 hybrid, the Fields and Song approach, where we have a DNA binding domain, sometimes Gal4, sometimes other DNA binding domains, LexA, fused to a bait protein, the protein that you want to understand who it interacts with, and then a library usually of praise that are designed and fused to an activation domain. And when this DNA binding domain and this activation domain are proximal to one another on a promoter and reporter vector, they interact, recruit the basal machinery, and activate transcription. And a couple of modifications to this method have made it really, really robust. And so now it's possible to do these types of studies either in liquid form using arrays or sequencing as an output, and be able to do these studies at genome scale in a sort of binary fashion. So yeast is already done. All the genes in yeast have been tested against all the other genes in yeast. And that's happened for other model organisms and even with human proteins where you have a relatively large number of entities, say 22,000 proteins times 22,000. There are groups like Mark Vidal's group at Dana-Farber that is approximately 1/3 or even 50% done with that interaction network. So some of the advantages of the yeast-2 hybrid is that it's really amenable to genome scale science. While n squared is a big number, it's finite and within reach for modern automation. And because you have this, you're taking advantage of this endogenous transcriptional process. You have really good signal amplification and good sensitivity. There are also downsides to the yeast-2 hybrid. Do these interactions actually occur in vivo? Maybe those proteins aren't expressed at the same time, so they might not. When they are observed out of context. So you could have interactions that occur in yeast but don't occur in a human cell, for example, because there's another higher affinity partner for one of the two factors. And of course, there's also the possibility for false negatives. There could be a human protein or gene that's missing in yeast that bridges two proteins, and of course, you would not read that out in the yeast system unless you had that protein. So here's the other sort of important and highly used approach, which is the IP mass spectrometry approach. In this approach, bait proteins are tagged with an affinity tag, for example, an HA tag or a MIC tag, and these tags are usually protein fusions are usually transfected into cells, and in this case, we're going to use the His system, and after binding to the endogenous proteins, this His tag is pulled down with a solid matrix, washed, and then the proteins are eluted off and trypsinogenized before mass spectrometry, and you solve the IDs of all the proteins that are in this complex using mass spectrometry and these searching algorithms. And in that way, you can take each bait and try to find as many proteins as it interacts with in a larger complex. So some of the advantages here are it's a native cellular context. In the case of putting it into a cell that contains that protein, because it can be in human cells, post-translational modifications can be maintained and available. For example, yeast don't have any tyrosine kinases, so if a particular interaction requires a tyrosine phosphorylation event, that may not read out properly in yeast. And it could be also more quantitative than the two hybrid, although I think there's some controversy over whether or not that's true. There are also disadvantages to the IP mass spectrometry method. First, it isn't all that sensitive. There's a high false negative rate. Second, it's not a genetic screen, so it is in principle harder to do in a high throughput fashion with respect to genetic screens. And the tags themselves can be sticky. And so some methods have been developed to sort of combat that, tandem affinity tags or TAP tags, where you have not just one tag, but you have multiple tags that you use to sort of cut down the noise. So question 3, true or false? Unlike two hybrid, IP mass spec allows for bridging interactions. That's actually false. Two hybrid can allow for bridging interactions provided the bridging protein is present. So like the example in yeast, if there's a yeast ortholog of a protein that bridges two proteins and it's present, it can allow it. Or if you did your two hybrid in a human cell, you could have a bridging interaction form. It doesn't preclude bridging interactions. It's just that IP mass spec better accommodates them in many cases. All right, so we've talked about the methods, but what have we learned from all these interaction studies? Well, a lot. And here's some early work where a couple of really interesting properties were identified. So here's a depiction of a whole organism interaction map, in this case a human interaction map. And some of the ideas that came from analyzing these networks early on, this is work from Zoltan Opvai's and Laszlo Barabasi's lab in 2001, where they described a couple of properties. One, that these networks were scale-free, and what that means is that there's relatively few genes that are highly connected and many genes that have relatively few connections. And if you plot it out in log space, you can see that these interactions follow a power law. And a second very interesting observation that came from these studies relatively early on and enabled by the large-scale reverse genetic studies, in this case the knockout panel, was that proteins or nodes that are central in these networks are more likely to be lethal when they're knocked out than peripheral nodes or proteins. So that's this concept of centrality and lethality. There are other concepts as well, and so I'll just describe several of those concepts on this one simple slide. So imagine that these are all these circles here and here are proteins or nodes in a network, and that the edges are different types of interactions. So this edge, all these edges in this particular map could be, for example, protein-protein interactions. So we have nodes and edges. This would be called sort of a hub node, and that means that it's not just connected to one particular target, but rather connected to 1, 2, 3, 4, 5 different other nodes. So it's a hub node, and it has a node degree. In this case, it's the number of interactions would be 1, 2, 3, 4, 5. So you can talk about the degree to which a node is connected as node degree. There's also a concept here of centrality. This is a really simple network. So in this case, you would say that A and C are more central, and B is more central than D here, which is out on the periphery. And another concept is called betweenness. In this case, this node here, B, is between two highly connected nodes A and C. These two highly connected proteins, and so this is betweenness. And if you're trying to draw a map from this particular protein here to this particular protein here, you get there faster by going through B, the shortest path here. Others have done some more sort of sequence-based, can combine sequence-based approaches with looking at these sort of generic properties. Here's a couple, a domain-domain interaction, where in this case, in this particular interaction network, these two interacting proteins share a similar domain. This is a homotypic interaction, and these properties, the domains tend to be a little bit longer. The interactions are higher affinity and more stable, and they give several examples here: BTB pos or RAS or card domains. And over here, here's a domain linear motif. So here the motifs range from are smaller, from between 3 and 10 amino acids. They're much lower affinity. Now the affinity is around a micromolar KD. They can be transient interactions, and here are several examples of these types of interactions here, like the SH3P, XXP, or the EVH1 motif. So the type of interactions in a network can differ, and those differences can sometimes predict the biochemical properties. There's another concept called party hubs and date hubs. So interaction information can be integrated with co-expression. So you can take one of these maps of interactions, for example, from the yeast to hybrid, or from from IP mass spectrometry, and you can integrate that with expression data, for example, from arrays. And when people have done those types of studies, they've described party hubs where most of the components are in the same place at the same time. For example, you could have a group of interactions that are all expressed in liver and during adult livers when they occur. And you can also have date hubs, which tend to have relatively low co-expression and occur in a particular time. For example, they occur during one stage of the cell cycle. So those are the concepts of party hubs and date hubs. Question number 4. The following concepts are true in protein interaction networks. A, they are scale-free. B, central proteins tend to be lethal when knocked out compared to more peripheral players. C, date hubs have high co-expression. D, all of the above, and E, only A and B. And of course, the answer here is E, A, and B, and that's because date hubs have relatively low co-expression. They're occurring in a sort of transient fashion, and the example I gave was at one stage in the cell cycle, whereas the party hubs tend to have high co-expression. Despite the strength of the protein interaction methods, there are also weaknesses to these approaches. They're not super easy and scalable, and so that makes it impractical to construct interaction maps of of different times. For example, what are the interactions at embryonic day 8 in a mouse, or in different places? What are all the interactions that are occurring within the suprachiasmatic nucleus of the hypothalamus, or in a different disease state? You can't very practically do all of your interaction studies from cells from different individuals, for example, because it takes a lot of time and resources to conduct these studies. I want to sort of finish off by reiterating something I mentioned earlier, which is that although we focused on protein-protein interactions in this discussion, you can basically integrate many types of different interactions. In this particular network, which is work from our lab, we have integrated protein-protein interactions, which are depicted by black edges here, but also phosphorylation reactions, which are depicted here in blue. Transactivation relationships. So these are all defined in green, where we have, for example, clock and BMAL activating Cry1, Cry2, and PER1 and PER2 and PER3. And also transrepression, where we have repressors like Cry1, which actually repress its own transcription. And all this information has been combined in a sort of organic layout and have this sort of described model. So final slide, there's going to be a lot more systems biology to come. As the technology develops, we'll be able to enumerate interactions, metabolites, modifications, many things that we haven't incorporated into these models to date. Increasingly, these studies will be kinetic, so instead of just occurring at one time point, we'll have more dynamic data coming in. And the models that will come from these large-scale screens will not only describe what happened, but also be predictive. As we learn more about systems, we'll be able to predict them better. That's the promise of systems biology.